The breast cancer cell spheroids described herein will allow researchers to investigate molecular mechanisms of cancer cell endothelial cell interactions that are involved in proliferation and metastasis of cancer cells. The main advantage of this 3D model is that it provides a more realistic cellular arrangement that improves the reliability of inferences regarding the pathophysiology and treatment of breast cancer. Demonstrating the procedure will be Giovanna Azzarito, a PhD student from my laboratory.
Begin by treating or transfecting the plated cells for 24 hours or as desired. For identifying cell distribution in spheroid, wash the cells with 1 milliliter of Hank's balanced salt solution, or HBSS, containing calcium and magnesium, and stain HUVEX using 0.5 micrograms per milliliter blue dye, and MCF7 cells using 0.5 micromolar green dye, diluted in the respective growth medium. Place the plate into a 37 degrees Celsius and 5% CO2 incubator for 30 to 40 minutes. Wash the cells with 1 milliliter of HBSS without calcium and magnesium. Then add 1 milliliter of enzymatic detachment reagent, which is 0.25% trypsin for HUVEC and 0.5% trypsin for MCF7, to each round dish using a P1000 pipette. Collect each cell type separately in a 5 milliliter round-bottom polystyrene tube, and add 1 milliliter of 10% fetal calf serum, or FCS medium, using a P1000 pipette to stop the enzymatic reaction.